if we were in a lab, what would the lab technician be doing? You know I've had guests on, they've done advanced cell spectrometry. Sometimes people are counting cells under a microscope. What are you guys looking at in the biopsy?
Yeah, those are all great techniques. We don't look at the cells. So what we do is we take the cells, we get rid of them. So we spin the tube really fast, create some artificial gravity to get the cells at, and we're left with a cell-free component that's called plasma. We take that plasma and we do some chemistry on it that grabs onto DNA and lets everything else go away. And that's DNA isolation or nucleic acid isolation. So now we're left with DNA. Some of it could just be normal DNA. So regular cells in your body are dividing, multiplying, and dying, and some of that DNA gets left in there. So just the fact that there's DNA doesn't mean that there's cancer. We've got to do some more testing to determine, is this DNA coming from cancer or from healthy cells? And what we do in the laboratory is a chemistry called bisulfite conversion. It's a chemical process that changes DNA based on whether or not it's methylated. And then we have some techniques that are similar to PCR that everyone's familiar with from COVID testing, where we amplify, we make extra copies of DNA that looks like it's coming from cancer. We're able to detect that then. We've got some computer software that analyzes that. We use some machine learning to help develop that and then make a determination of, hey, do we think that this is cancer or not?
Okay. So PCR is polymer chain reaction for anybody. Is that correct?
Polymerase chain reaction.
Polymerase chain reaction.
There you go.
And then we talked about methylation pathways. Could you explain? Because sometimes we'll say a term and there's an assumption that everybody knows what we're talking about. Now, I have a supplement in my cabinet that is methylation support from Thorne because I want help methylating. I've looked at my genetics and it says, Freddie, you might want this in your longevity stack. Can you describe to us the process of methylation and why it's important?
Absolutely. So you've got different cells in your body that do different jobs, right? But you only have one genome. So the genome, the DNA, you got one copy of your genes from mom, one from dad. That's kind of a fixed static thing. And it's the instructions for all of the protein machinery in your body, all the enzymes in your whole body. But a neuron doesn't need the same machinery that a skin cell needs, right? They've got very different jobs, but they have the same blueprints. Epigenetics is the study of how does the body turn genes on and off in a healthy way, and how are genes turned on and off in disease as well. So aging is likely a methylation-related disease. That's why those supplements make sense scientifically. And then cancer is also a methylation epigenetic-based disease. So cancer, you get genetic changes, but also that same methylation framework that changes gene expression in a healthy way in different parts of your body. It can use some of the machinery that normally isn't used by a cell. It can turn genes on, turn genes off in a way that changes the behavior of those cells and helps them develop cancer. Methylation is one of those mechanisms. Methyl is a little carbon that can get attached to a DNA. In humans, we talk about 5 cytomethylation. So cytosine becomes methylated at a specific site there. And usually what happens is that methyl group is added in a part of the gene that doesn't get turned into protein, but it's important for the expression of that. So do we want the machinery to lay down there and turn that gene into functional protein or not? And so it's just flipping switches on and off that allows the cancer cell to use some of the machinery that's useful and helpful otherwise, right? But in this context of cancer is dangerous.
Yeah. So what's happening in the body, essentially, we're getting this abnormal methylation pathway.
Yeah. So it's an abnormal, right? So the methylation that is normally there and healthy, right, is changing. And so we're getting activation of genes that may normally be associated with growth and repair, which is a good thing, right? Growth, but out of control growth is bad, and cancer has out of control growth, right? And so some of the same methylation things that can be useful during that healing process, healing is done, and now it's causing disease.
Yeah.
Yeah.
I want to just step off the trail for a second and just,
Sure.
what's always amazed me about the human body is we talk about the genes and how many, we have a blueprint for cells. And then there's so many more protein expressions than we have like the blueprint for. That has always been one thing that I'm like, it's hard to imagine a system that's more intelligent than the human body. Right? It's just got to do all these incredible things from this small amount of, how many protein expressions are there? Like 140,000? It's a lot.
We have about 20,000 genes total, right? So that's coding things, but those can be recombined and modified in different ways. So we call them isoforms, where, in one biological context, the gene will do one thing, and in another context, it'll be cut up differently, twisted around differently, and do something else. So you're absolutely right. It's like it's almost like a compression algorithm, right? Where you've got all this data and then it's been compressed down into this tiny footprint. It is beautiful, absolutely.
Yeah.